Hey, hello everyone. This is video number 5 for Module 7. And in this video, we'll start talking about how genes are regulated in eukaryotes. Let's take a look at the outline here. And so what I'd like to cover in this video is a general description of gene regulation in eukaryotes. Some features are similar to prokaryotes and some are different. And then we're going to focus in on transcription regulation at the level of initiation of transcription and define what are the main factors that are involved both at the DNA level and protein factors as well. Okay, so let's take a look. This first slide shows this is similar between prokaryotes and eukaryotes, just that there are multiple levels at which genes can be regulated, and that is at the level of transcription, initiation, elongation, termination. In eukaryotes is another step which involves processing of the primary RNA transcript. That's another level that can be regulated. And in eukaryotes, there are different compartments for transcription and translation, so that can be regulated as well. Transport of RNA, mRNA out of the nucleus to the cytoplasm. And then once RNA gets to the cytoplasm, stability, rate of translation, protein stability, and protein modification are all mechanisms that can and do regulate expression of proteins encoded by genes. We are going to focus, like we did for prokaryotes, at the very beginning of this process. This step, the initiation of transcription. Okay? And just to remind you also that for eukaryotes being mostly multicellular, the aim of studying gene regulation in the past has been mostly geared toward understanding how different cells in a multicellular organism come to adopt different fates, how they differentiate from each other, and how they carry out specific functions in that differentiated state. Basically, how a muscle cell develops in one case, and how a neuron can develop using the same genetic information. So one thing I want to point out about transcription in eukaryotes that's different from prokaryotes is this point right here. There are 3 different RNA polymerases in eukaryotes. RNA polymerase 1 transcribes the genes for ribosomal RNAs. These are non-coding RNAs, part of the ribosome. RNA polymerase 3 transcribes genes for tRNAs and other small RNAs, also non-coding RNAs. They take part in translation. We are going to focus on RNA polymerase 2 transcribed genes. These are the protein coding genes. Okay? So, what's involved in regulating transcription in eukaryotes? So there's similarities between eukaryotes and prokaryotes. There are cis-acting DNA elements and trans-acting proteins. The cast of characters is a little bit different. So I prepared a short video to go through those different factors. And so let's take a look at that now. All right, let's talk about eukaryotic gene regulation. And we are going to focus in on regulation at the level of transcription initiation. The most common point of regulation for gene expression. And what are the main things involved in regulating gene expression in eukaryotes? 2 main things, cis-acting elements and trans-acting factors. Cis-acting elements are DNA sequences. And these are going to regulate the gene that they are closely associated with. Okay? There are 2 main types, promoter and enhancer. The promoter is going to be attracting RNA polymerase to start the transcription. It's going to set a basal rate of transcription. the enhancer is going to modify that basal rate either up or down, okay? These cis-acting elements bind to transacting factors, which are proteins. These transacting factors are made by other genes in the genome, and they can diffuse through the nucleus to bind to these transcription elements that are regulating a given gene, okay? So cis-acting elements are regulating only the gene that they're closely associated in DNA sequence with. Transacting factors can regulate any gene on any chromosome because they're able to diffuse through the nucleus and latch onto and therefore regulate that gene, okay? Three main types that we'll talk about: the basal factors. They're involved in binding to the promoter, and they're going to set the basal rate of transcription. And then there are the activators and repressors, and these are going to bind to enhancers, and are either going to turn up that basal rate or turn down that basal rate. Okay, so those are the main elements involved in regulating gene expression at the level of initiation of transcription. Okay, so this slide is just showing that information. Cis-acting elements are the promoter, and the promoter here right next to the start of transcription. This is going to be attracting the RNA polymerase, so it knows where to start. And the enhancer shown here, the enhancer is going to somehow influence, for now let's keep it vague, influence the activity of RNA polymerase. It can either increase it or decrease it. Okay, so those are the two main cis-acting elements, promoter and enhancer. Now, I prepared short videos to describe each of those in a little bit more detail, so let's start with one for the promoter first. All right, let's talk about a eukaryotic promoter. What is the structure of a eukaryotic gene promoter? All right, so eukaryotic promoter, here's some characteristics. There's one promoter per gene only, and the location of the promoter is right next to the transcription start site. Remember, the function of promoters in prokaryotic genes is going to guide the RNA polymerase to the start site of transcription. Similar spacing in eukaryotic promoters. So if this blue line is a gene and here is the start of transcription for this gene, the promoter is going to be right next to that. Okay, it's not a very big stretch of DNA, usually 50 bases or so, 60. But here's one thing: in a eukaryotic promoter, it always has a consensus sequence. Consensus sequence means this is conserved amongst all eukaryotic gene promoters, common element of all gene promoters. And the consensus sequence is called a Tata box, basically TAA, embedded about 30 base pairs upstream from the start of transcription, Tata box, okay? Now, the eukaryotic promoter is going to be involved in binding to transcription, transacting protein factors. And the general term for transacting factors that are involved in regulating transcription is transcription factors, okay? So there's a couple of factors these are called the basal factors that are going to be involved in binding to the promoter or the promoter itself or to proteins that have already bound to the promoter. So the basal factors are one called the TBP, that stands for the Tata binding protein. So this protein comes along, TBP directly binds to DNA at the Tata box, okay? That's one basal factor. Second basal factor or set of factors are called TAFs. Those are Tata binding protein associated factors, TAFs. They don't bind to the DNA directly. They do bind to Tata binding protein if it's bound to DNA, bound to the Tata box. If it's not bound to DNA, they won't bind, okay? So, Tata box, TBP binds first, and then TAFs bind. Okay, once you've assembled these two, TBP and TAFs, then that's going to attract the big the big molecule, the RNA polymerase, to the promoter. All right, you can think of it as bait. You've set the trap, there's the bait, and the RNA polymerase is going to latch on. All right, now you've set RNA polymerase 2 right next to the start of transcription, and then it's going to start doing its job transcribing this gene. One thing to think about though, these are called the basal transcription factors. Basal transcription factors bind the promoter. And they set up RNA polymerase to transcribe at a very low rate, the basal rate, okay? Then the enhancer comes in to either increase that over the basal rate or decrease it, push it close to 0 transcription, okay? So this is all involved in setting up basal rate of transcription through these basal transcription factors. Let's talk about the enhancer next. Okay, I also prepared a short video for the enhancer, so let's take a look at that. All right, let's talk about enhancers next. Enhancers are cis-acting DNA elements. And here are some features of enhancers. So this is a diagram here on the top. Blue line is a chromosome segment of DNA. And let's say this has a gene with a coding region here shown separated into 2 exons and an intron in between. Here is the start site of transcription. +1 here marks where RNA polymerase is going to start to transcribe this gene. And here's the promoter right next to the start site of transcription. All right, how about enhancers? So unlike promoters, there's only one promoter per gene. There can be multiple enhancers. So it's not necessary. There can be a single enhancer, 2, 3, 4. There can be multiple enhancers, okay? Unlike promoters where there's a consensus sequence, the Tata box, there is no consensus sequence for an enhancer. They vary a lot in their sequences. So there's nothing that's conserved among all enhancers. Okay, the location of the enhancer, unlike the promoter, which is always right next to the start of transcription, enhancers can be anywhere around this gene. They can be downstream, this would be the 3' end of the RNA. They could be upstream, so on the other side of the promoter. They could even be in an intron of the gene. And they can be really distant upstream. So this is a hash mark here indicating a break in the chromosome. There can be thousands of base pairs away, okay? So there's not as much restriction on the number or the place that enhancers can be, unlike the situation for promoters, okay? Finally, enhancers are going to be binding to transcription factors that are either going to upregulate transcription from this promoter, the case for activators, or downregulate it, the case for repressors, okay? So those are the main features of enhancers. Okay, just a couple of slides to illustrate those principles once again. So here are the 2 cis-acting elements, promoter right next to the start of transcription, and enhancer. In this case, it's on the promoter side of the gene, but let's say it's further away. These are going to be involved in the promoter binding to basal transcription factors, Tata binding protein and Tata associated factors, TBP and TAF. And then those are going to attract the RNA polymerase. For a protein coding gene, this would be RNA polymerase 2. The enhancer is going to be involved in binding to transcription factors that can be either activators or repressors. If they're activators, these activators at the enhancer are somehow going to influence transcription rate from the promoter in the positive direction, increase rate of transcription. If they're repressors, they're going to turn this down, basically shut it off, okay? So that's kind of a summary of the cis-acting elements and the transacting factors. And this slide is just showing the basal factors binding to the promoter through the TATA box. TATA binding protein binds it directly. TAFs latch onto the TBP, and then that altogether attracts the RNA polymerase II to the initiation complex. All right, I think I'll stop this video here, and in the next video I'll talk about activators and repressors, how they work through binding to enhancers, and how can enhancers distant from a promoter influence the rate of transcription at that promoter. See ya.